this is where things get interesting. So we talked about all the different parts of the innate or non-specific immune system, those things that you're born with, intact skin, different chemical protections, right? Remember, they're non-specific. They don't care who the invader is, they're going to attack. Now we're getting to adaptive, or what's also called specific immunity. This is the true immune system, true immunity. These defenses are very picky. This is what lets you differentiate between influenza H1N1 or influenza H3N2, right? Your body knows what you have been exposed to before, right? So this adaptive immunity, this is key. This is not innate. These defenses are not present at birth. The cells, the lymphocytes that we're going to talk about, are things that you are born with, but they only develop and create this vast army in response to an exposure to a pathogen. Okay, so sometimes you might also see this referred to as acquired immunity, right? So it's adaptive, it's specific, and it's acquired. Now, what they're showing you here, right, is as we talk about adaptive immunity, there's a few different ways to get this, right? You can either get it actively, immunity either actively or passively. What we mean by actively is that we are developing antibodies in response to antigen exposure, right? Or I guess it doesn't just have to be antibodies. We're developing this army of lymphocytes that are going to respond after exposure to that antigen. Now, this can happen naturally. Someone coughs on you in class, right? You get exposed to those antigens and your body mounts this response, right? So after exposure to antigens in the environment, or artificially, this would be vaccination. So in vaccination, the whole point is to get a small dose of an antigen, right? Maybe a surface protein, maybe an injured or dead virus, bacteria, something that's going to give you exposure to the antigen. And heck, now with these mRNA vaccines, right, we're just getting exposed to some of this messenger RNA, right? And we can create proteins and then respond to them, right? So that would be, both of these are active immunity, but one is natural, one is artificial. On passive immunity, basically what we're looking at here is that you're receiving antibodies that have already been created elsewhere. Now, this absolutely happens naturally from mother to baby. This can either be antibodies that are crossing the placenta, right, during pregnancy, and then also after childbirth, mom is passing antibodies to the baby through breast milk. This is one of the reasons that breastfeeding can be super important. It helps to give the baby some defenses until it's a little bit older and can start responding on its own. Obviously, it's not necessary for life, but it is helpful, and you'll see babies get sick less often if they're breastfed. Artificial acquired passive immunity, that's a mouthful, is when a patient is given preformed antibodies. You might have heard with COVID, they were talking about monoclonal antibodies, right? They're produced somewhere else. Sometimes we use things like rabbits. So you can expose a rabbit to rabies. It will start to produce antibodies against the virus, and then you could collect those antibodies and inject them into a human patient to keep them from dying if they're exposed to rabies, right? But the human immune system, by the time it catches on to rabies, the patient dies, right? So you want to get those antibodies from somewhere else. Or maybe you've heard they were doing this with Ebola, right? There's no treatment, and they were calling it convalescent therapy. So people who were recovering from Ebola, right, they're known to have the antibodies. You could collect those antibodies and give them to someone who's sick with Ebola. Now that kind of went poorly because people started hearing like the blood of people recovering is useful in preventing Ebola, but if not done well, you really shouldn't just be transferring whole blood, and there's viruses, right? That part of Africa, there's a lot of HIV, anyway, but it can be done well as well. Okay, so active, natural and artificial, passive, natural, and artificial. So we have really 4 different ways that we can get this immunity. And here's a key thing, when you think about immunity, what does it mean to be immune to something? It means you're not going to get sick, right? The first time you're exposed to that antigen in the environment, you do get sick. But if everything works well, right, you create the antibodies, you create all the lymphocytes that you need, next exposure, you are immune. You don't get sick. Vaccination, right? Your body is going to respond, create the army. If you get exposed in the natural environment, you shouldn't get sick, right? Mom's giving you antibodies. Someone who's recovered, or a rabbit who has died, is giving you antibodies. Okay, so those are our forms of adaptive or specific immunity. Now, all of these, right, when we're in the specific or adaptive immune system, all of these are going to be based off of lymphocytes. Right? So this is the cell of the adaptive specific immune system, right, are the lymphocytes. Now there's a couple different categories that you read about, right? There are T cells, and there are a huge variety of T cells, okay? So we'll talk a bit about cytotoxic T cells. These are lymphocytes that can sense infected cells or cells that have a mutation, maybe a cancerous cell, and they actually go in and kill that cell, right? It's your own cell, it's infected, but these cytotoxic T cells are like, you are taking one for the team, we're getting rid of you before this illness can spread, right? So when we talk about T cells, particularly these ones, you might hear people talk about cell-mediated immunity, right? And it's the T cells really that are doing this. There's also helper T cells, and these are the linchpin of the immune system. The rest of it fails to work without these helper T cells. So we'll go through, we'll look at how cells are kind of communicating with each other in this process, but it's really the T cells that are going to send out cytokines, chemical messages that go to things like cytotoxic T cells and be like, yeah, go get them, right? Or to B lymphocytes, which we'll talk about in a second, and say, yeah, it's time to make antibodies. Nobody does the work unless the helper T cell encourages them to, right? So they help everyone, or you can think of them as like the cheerleaders. They're like spurring everyone on to action, right? But they absolutely are the keystone, the central piece. And so that's actually why HIV is so damaging. HIV takes out these helper T cells. And even then, if your cytotoxic T cells or your B cells are fine, they're not getting the messages, right? And those individuals typically are going to die from a secondary infection because the immune system doesn't respond without helper T cells, right? There's some others. There are suppressor. I will say these two, cytotoxic and helper, are the ones we'll talk about the most. There are suppressor T cells. These are ones that kind of calm the immune system, be like, okay, we got the bad guy, we're done, everybody chill out, right? So there's some talk about like autoimmune diseases. Some of these suppressor T cells may not be calming the response, right? And that could lead to something like autoimmunity, right? There are others, again, these are going to be the two main ones that we focus on, right? We also have B cells, right? And again, the other way to say this is T lymphocyte and B lymphocyte. But very common to call them T cells, B cells. Anyone in the medical profession will know what you're talking about. Now, B cells, they do not do cell-mediated immunity. They do what's called humoral mediated immunity. This is is a really old term, right? So this goes back to when we talked about, not we, but medicine or science talked about the humors of the body, right? As someone, I don't know, it's the fluids in the body, right? And they talk about if someone's too hot or this or that. The humors, the fluids. Anyway, maybe a better way to think about this is probably a more up-to-date term of like antibody-mediated immunity, because those antibodies are floating around in the plasma, the fluids, right, that they're talking about here, right? So antibodies, these are proteins that are produced by B cells, and very specific B cells. B cells are actually going to convert themselves into what's called a plasma cell when called upon to make antibodies. So a plasma cell is a B lymphocyte that's producing antibodies. Sorry, your notes just got really confusing there, right? And again, these antibodies, they're proteins, and we'll look at a variety of different ways that they can attack pathogens and help keep us from getting sick, help give us immunity. Now, all the lymphocytes that we're talking about are going to originate in the bone marrow. Just like we saw in hematopoiesis, we focused on the red blood cells, but we know that there were white blood cells being produced as well, right? We can see now that lymphoid lineage. Remember, all the macrophages and neutrophils, eosinophils, red blood cells, all that stuff was coming from a myeloid stem cell lineage. Remember, lymphocytes all come from this lymphoid stem cell. So all of our lymphocytes, T cells, B cells, come from the bone marrow, from these lymphoid stem cells. And then we're going to see things start to diverge. So for example, right, B cells, right, they came from the bone marrow, B cells, and they are going to move into circulation, be making those antibodies, right? The NK cells, remember, those natural killer cells are actually part of the innate immune system. They're doing that immune surveillance, but they're that kind of strange one, right? They do come from that lymphoid stem cell lineage as well, right? Doing immune surveillance, which they recognize anything that's foreign. So that was our innate or non-specific immune system, right? We then also have some of our lymphocytes that are going to migrate to the thymus and become T cells, right? So everyone originates in the bone marrow. B cells go straight out into the blood and the lymph nodes and that sort of thing. T cells are going to go to the thymus first, and they go there for some special training. So the thing to keep in mind is with all of our lymphocytes, they all work on having antigen receptors, right? So a couple key words: antigen and antigen receptor. Right? Where do we find the antigen? The antigen, the definition is really just any protein that stimulates an immune response, but typically what we're talking about is like a surface protein, like I'm drawing here on bacteria, little red triangles, antigens. They're going to trigger an immune response. And they do that because all of our lymphocytes, B cells, T cells, whatever branch, right, have surface proteins with variable shapes. Now, each lymphocyte only has one shape. All right, so I can draw a few different lymphocytes here and give their antigen receptors different shapes. Now, this is fascinating. I hope you talked about this maybe some more in microbiology, but these antigen receptors come in a variety of shapes. In fact, you have lymphocytes with receptors for things that don't exist, or Martian dust maybe, right? Because they're actually made quite randomly. When those antigen receptors, these proteins, are being built, the DNA kind of gets, I don't want to say scrambled, but you kind of pick and choose random pieces of DNA and then read it and say, ah, this is the protein we're going to build, right? So this is a little different than the way you normally think of DNA being made. So you have all these kind of possibilities encoded in the DNA, and then when you go to build a receptor, you pick random things and you put it together. Now I'll draw these as shapes like triangles and squares. It's much more complicated, obviously, right? Because it needs to perfectly fit something on the surface of a microbe, right? And it's not a triangle, it's something that can differentiate one strain of influenza from another or one strain of E. coli from another. But my artistic skills end here. So antigen receptors, surface proteins that attach to antigens and really are the basis of our ability to respond, right? So this lymphocyte, right, can find this strain of E. coli. This guy would do nothing, right? He can't attach, and that's necessary in order for the immune system to work. Okay, now we can take it up a notch from there because this chart is talking about MHC proteins. Okay, everybody good? Antigen receptor. Okay, sneak peek. If you're talking about a B cell, its antigen receptor, when it becomes a plasma cell and starts producing antibodies, they have the exact same shape as that cell's antigen receptors. The same protein is built, it's essentially just dumped into the bloodstream. Okay. Now, another important protein in this communication are what are called MHC proteins. So all cells in the body, right, have, and I shouldn't say all, there are a couple exceptions, most cells in the body have a surface protein called MHC1. And let's just say all cells to make your life easier. It's like red blood cells don't, but okay. So MHC stands for major histocompatibility complex. Okay. Sometimes, think about this. Remember histology, the study of tissues. So this is tissue compatibility proteins. As doctors started doing organ transplants, they realized it was more than just matching blood types. There was something else that caused tissue rejection, and it was actually how well these MHC proteins are matched. Okay? So what this MHC protein, this MHC1 is, right? It is a protein. Now remember, every protein that's built, it's based off of the DNA code. So in the nucleus, right, you've got DNA that tells you how to produce MHC1, right? You make your little strand of RNA, it goes out to a ribosome. I realize this is tiny. It goes out to a ribosome, and you start building the protein. Transcription, translation, everybody with me? Okay, this MHC1 protein, oh goodness, let's try to draw it kind of like they did. Right? It's this big protein, and it is going to go to the surface of the cell, but its job is to grab a sample of whatever protein is right? That's what they're trying to show you here. Whatever protein is being made by the cell. So I'll just make that, oh goodness, a red rectangle, right? And so this MHC1 migrates to the surface of the cell. I will try to draw it again. You can draw it however you want. It's actually pretty good for me, okay? And it's still holding, oh my goodness. Now I'm struggling on the rectangle. It's holding this on the surface. It is holding this on the surface of the cell, right, in order to be checked. Checked by whom? MHC1 proteins always get checked by cytotoxic T cells. Recall we said cytotoxic T cells, their whole job is to look for a virus or a cancer and then destroy an infected cell. So let's think about this. Now in this example, I just had it pick up a protein that's self, that's me, right? That's normal, because cells are producing proteins all the time. Maybe it's a hormone, maybe it's a sodium-potassium pump, maybe it's some other enzyme. But if I put this on the surface, my cytotoxic T cells come around checking MHC1s. Now, our cytotoxic T cell has an antigen receptor, and I very specifically made one that didn't match. This is key to the function of the immune system. You should not produce any lymphocytes with an antigen receptor that matches your own proteins, right? That would trigger an autoimmune disease. You would start attacking yourself. In fact, that's a really important thing that happens in the thymus. That's the specialized training. In the thymus, we screen all the lymphocytes. So if you did happen to randomly produce one that would fit my red rectangle, my sodium-potassium pump, or whatever it is, in the thymus, if it reacts, it gets destroyed, right? We only want to attack foreign antigens. So this cytotoxic T cell comes up here. Cytotoxic T cells, you will also sometimes see them called CD8 cells. And this stands for cluster of differentiation. And yes, you're thinking this is a total cluster, where it's just this special protein. Now my drawing is really gonna stink, right? That kind of shakes hands. That's really big protein. That kind of shakes hands with MHC1. So CD8 cells, cytotoxic T cells, always check MHC1s, and if the protein is self, sorry, so this is the CD8 protein. If, I'll write that, CD8 protein attaches to MHC1, right? They're shaking hands. The antigen that's on the surface, my little red rectangle, if it doesn't fit the antigen receptor, what does this guy do? He moves on, right? Not a big deal. Oh, that looks normal, right? If the antigen receptor matches the antigen, well then we've found an invader, right? So keep in mind, your DNA builds your proteins, but if you had a virus, how do viruses work? They take over your machinery and say, hey, ribosome, psst, time to build influenza virus, right? The spike proteins and this sort of stuff. And then your MHC1 would hold that flu virus to the surface. If your cytotoxic T cell found that protein, right, if it was a triangle in this case, this guy, your cytotoxic T cell, whips out a gun and shoots, that's a little handgun, shoots the infected cell, right? Same thing, if you have a mutation in your DNA, if you have cancer, you would produce an abnormal protein. If a cytotoxic T cell shakes hands with the MHC1 and your antigen receptor matches the antigen, boom, you're going to destroy it, right? So they actually use perforins, which we've heard before, in order to destroy an infected, a virally infected or cancerous cell. See what I mean? This stuff gets cool. You should totally take immunology someday, right? Because It gets way more complicated than this. I realize this seems complicated. I'm trying to kind of narrow it down. Okay, now, a couple things to keep in mind. What they're trying to show you on this graph, right, is CD8 cells check MHC1 proteins, right? Cytotoxic T cells attack and destroy if this cell is showing a non-self protein, a foreign protein, right? You also then will get what are called memory T cells. So when a lymphocyte actually finds an antigen that it matches, I'm going to skip ahead a couple here, you get what's called clonal selection and differentiation. So notice what they're trying to show here, right? Lots of different antigen receptor shapes. This is where I get my art suggestions, right? Here's my foreign antigen. There is only one lymphocyte, and this is totally true. The first time you're exposed to an antigen, there might be one lymphocyte in your body with the correct receptor shape, right? And he's just been floating around, thinking someday I might run into mono, right? And then his day comes. It's the chosen one. This is the only lymphocyte in your body that can protect you, right? So he's selected, and then we start cloning, right? Now they're showing here with B cells, but the exact same thing happens with T cells. That cytotoxic T cell that's the chosen one starts cloning itself, okay? So you build this vast army, thousands, right, of these cells. And very importantly, every time a lymphocyte goes through clonal selection, some of the cells that it leaves behind, well, kind of small, puny little things, they're for next time. These are memory cells, right? So they're pointing out here memory cytotoxic T cells. So we get clonal selection. You're the chosen one, right? And then differentiation, differentiation, but focus on the cloning, right? So you're selected, and then you make lots of copies. I'm probably off the screen. Lots of copies. We'll bring it back up here. And some of those copies are memory cytotoxic T cells, right? Next time you come into contact with that exact same antigen, now there's not one. Maybe it's been years since you've been exposed, but if there's 100, that clonal selection, that differentiation happens that much faster, and you can actually replicate lymphocytes faster than that pathogen can multiply itself, okay? So that is what's going on with these MHC1 cells. Sorry, and then they're just pointing out suppressor T cells again. After the infection is over, we need to calm this down, right? Again, they're pointing out the point of cytotoxic T cells is direct attack, right, and destroying wherever they find those antigens. The other side of this graph then is talking about MHC2 proteins. Now these are really only found on specialized cells. So we talk about them as antigen presenting cells, or APCs, antigen presenting cells. This includes B cells and macrophages. Macrophages are the big ones we're going to focus on. And then any little specialized macrophage. So if you remember in the integument, you had specialized cells called dendritic cells. Those are just macrophages that now live in the skin, right? Or the microglia in the brain, or Kupffer cells in the liver. Anyway, okay, macrophages is fine, right? So basically what happens is when a macrophage engulfs a bacteria, right, some foreign object, it then has that antigen inside itself. It becomes an antigen-presenting cell. It's still a macrophage. Okay, let's see if I can do this. So my macrophage finds a bacteria to engulf, right? And it's got all these little surface proteins, antigens on our bacterium. When those get brought into the cell, the macrophage is going to break it up and destroy it, right? And so we get all these little fragments of whatever the macrophage found. Along comes MHC2, right? Because your DNA in that cell tells you, here's my ribosome, how to make, I should make it look different, shouldn't I? They make it look exactly the same. It's really similar. It's an MHC2 protein. So I'm just not going to make it as complex, right? It's going to grab samples of proteins that have been ground up by the cell. So MHC2 does not attach to any self-proteins. It's only taking to the surface foreign proteins, right? Always antigens. So antigen-presenting cells, or macrophages, once they grind up a bacteria, the MHC2 carries a sample to the surface. Well, who are we trying to show that to? We are trying to show that to helper T cells. So along comes my helper T cell. Ooh, this one has, that's a circle. This one has, I know you can't tell it, but that has the correct antigen receptor shape, right? So this is my helper T cell, okay? And a helper T cell is sometimes called a CD4 cell, cluster of differentiation 4, because the weird protein that it has shakes hands with MHC2, right? So this big blue protein that I tried to draw always docks with MHC2, and then my antigen receptor swings into place and checks. Oh, hey, I match this one. That's not a good thing, right? If I match it, that means it's foreign and we're going to need to attack, right? Our helper T cell is not wanting to destroy the macrophage though, right? So this is not like a cytotoxic T cell whipping out some pistols and blowing this cell up, but it is going to start the process of activating B cells, right? So helper T cells, remember we said they put out all these kind of cytokines. It's going to find a B cell, and again, this gets a little more complicated, but it finds a B cell with that same antigen receptor shape, and it activates it. It says, you're the chosen one, right? And we do that same cloning that we talked about before, right? And in B lymphocytes, some of these cells are going to become huge protein-making factories, right? And they are going to start just cranking out billions of these proteins that look just like the antigen receptor. And when those proteins move into the blood, we call them antibodies. Good. And remember, this huge protein factory is just called a plasma cell. It's still a B lymphocyte, but it's specialized in making antibodies. We also make some little piddly ones called memory B cells to keep around for later. In fact, this helper T cell, once it comes in contact with its antigen, does the same thing. It clones itself, right? Might need more of me, and some of those clones are memory cells, right? So we get memory helper T cells, we get memory B cells, we get plasma cells, and we get antibodies. And it's the antibodies that are then going to be floating around the blood, right? And looking for any bacteria that they can attach to. Okay, so I just got a lot there. The MHC2 is always checked by helper T cells, CD4 cells. If the correct match is made between an antigen and an antigen receptor, we go ahead and activate B cells. In reality, you would also activate cytotoxic T cells, right? If this is a virus, some of it's already in cells. Anyway, those cytokines activate B cells, they go through clonal selection, plasma cells make antibodies, antibodies seek and destroy. Right? This is complex. This is kind of crazy that this is happening in your body all the time. And again, as long as you have memory cells, if you've witnessed this antigen before, you should be able to raise this army of lymphocytes before it can make you sick again. So the book has some really great images, frankly much better than what I just drew for you. I'd like to focus your attention, I think 2818 is a really nice look at antigen presentation. This one's showing MHC1, right? So putting abnormal proteins, well, self and abnormal on the surface. If it's abnormal, it's going to get attacked. Where'd I go? There we go. This one is trying to show a macrophage, right? It's breaking down that bacteria. It becomes an antigen presenting cell when those MHC2 proteins hold it to the surface. So those are really well drawn explanations of where those MHC proteins come from. In class, ask me about the t-shirt study, unless you've already heard that tangent. Okay, 2219 is a real nice kind of summary of some of the things I was just talking about. So here we have a cell that's infected, right? Picture a little virus inside. That MHC1 protein, right, is holding it on the surface. And here we see that CD8 or cytotoxic T cell checking it. Look at this zoomed in. This is way better than my drawing, right? So class 1 MHC holding an antigen on the surface, right? Our CD8 cell, right, here's the CD8 protein shaking hands with the MHC1, and this particular receptor matches the antigen, so we know it's foreign, right? Look at what happens when we recognize that it's foreign, right? Once we've recognized it, we start doing all the cloning we talked about, including of memory cells, and here's our close-up of our cytotoxic T cell using perforins, right, and other things, right, cytokines, lymphotoxins. Perforins are the big ones, shooting holes in that cell and getting it to lyse, right? So there's a nice summary of what cytotoxic T cells are doing. Okay, here we're looking at an antigen-presenting cell. So they have what class of MHC proteins? Antigen-presenting cells have MHC2 proteins on the surface, and so they're showing that holding an antigen. Our helper T cell, right, has a CD4 protein. That's how it shakes hands with the MHC2. Just gotta get this all laid out. We'll be all right. And then we can activate that. And again, chemical messages coming from the helper T cells get everybody else activated. We go through all the cloning and we keep memory cells. Memory cells are the key to immunity. Okay, figure 2223 is showing us how B cells really get romping here. So here we see our B lymphocyte, and it attaches to some antigens. And this is where I said B cells also have an MHC2 protein. Because remember, that B cell, even if it has the right antigen receptor, until a helper T cell tells it to differentiate, tells it to clone itself, tells it to make antibodies, it doesn't happen. So what they're trying to show you in this close-up is that it is, in fact, helper T cells. This is the CD4 protein again, shaking hands with MHC2. And if that antigen matches one on the helper T cell, that helper T cell starts sending out the cytokines, the chemical messages to encourage cloning, plasma cell differentiation, and the production of antibodies. Right? So this is B cells. They're making antibodies. Now, antibodies I do want you to understand like what they are, because I think a lot of times we just throw it out there and they kind of sound like they're magic. So antibodies, again, we said are proteins, and you don't need to know all these details about light chain, heavy chain, but they are the exact same shape as the antigen receptor on the lymphocyte that makes them. And what they're trying to show you here then is those antibodies docking on the antigens on the surface of this foreign invader. Okay? Now, antibodies, the other name for them is immunoglobulins. Right, so same thing, but when you see IgG, right, that's immunoglobulin G. It's an antibody. So there's different classes of antibodies, and I don't need you to know all of the details here, but let's just pick out a few things, right? So IgG, this is the largest, right, and it's the most prevalent, or numerous, right? It's 80% of the antibodies that we produce. This is also a really important one for passive immunity. IgG is able to cross the placenta. Right, and in fact, this gets into some of the hemolytic disease in the newborn, right? These anti-RH antibodies. We can talk about that more too if you want. Right, so that's really important, right? They're the largest, they're the most prevalent, for sure. IgE, you should associate with allergies. Because one of the things that IgE does is it binds to basophils and mast cells. And do you remember what they contain? Histamine. So when IgE binds to a basophil or a mast cell, it releases histamine, right? And this is going to be what's leading to that inflammation in the area, and that's essentially the allergic response. Okay. IgD, we don't really talk about a whole lot. I'm okay if you just know it exists. IgM is this crazy looking antibody. So this is a pentamer. It actually has 5 different binding sites. And so the cool thing with something like this, if you think about it, it can attack a bunch of different, say, bacteria at the same time, kind of clump them together. Okay? So that's the agglutination. What else was I going to say on this? Oh, this is the first antibody produced. And we'll look, these antibodies kind of come in waves. IgM is the first antibody, and it does circulate as a pentamer, right, a 5-sided cluster. Okay. IgA, so I'm out of room down there. IgA, this is a really common antibody to find on the respiratory mucosa. Okay. And so they're adding extra protection to those mucous membranes, right? They're not quite as hard, they're not dry like skin, and so those mucous membranes are a little more susceptible. And so we have this IgA that's basically on those surfaces to add extra protection. This is an interesting one. You can run into situations with immunity, right, where the entire immune system is failing, something like HIV, right, because of that helper T cell not working. But you can also have very minute parts of the immune system that aren't functioning properly. So for example, you can have what's called selective IgA deficiency, right? There are some individuals that just don't make IgA. I have a friend with this, and he ends up with pneumonia a few times a year, right? That respiratory surface isn't as well protected. Not ideal, but also not as bad as, say, being born with severe combined immunodeficiency where like nothing in your immune system works, right? So anyway, kind of interesting. So here's this graph. What they're showing you here, the primary response, this is the first time you are exposed to that antigen, to that pathogen, the illness, right? And here you see IgM coming on quick. IgG is a little bit slower, but it does become much more predominant, right? Notice the timeline here. From that point of infection, right, it's taking a week or two until those antibody levels rise. In the meantime, you're sick, right? Until these levels are up here, that's when you're snotty, coughing, you've got a fever, whatever, right? Now, the secondary response, this is any subsequent exposure to that same antigen. Look at this. You get exposed, those levels skyrocket because you have all the memory cells, the memory helper T cells, the memory cytotoxic T cells, the memory B cells, right? In this case, particularly those memory B cells very quickly can convert to plasma cells and start cranking out the antibodies. That virus doesn't stand a chance, right? You're gonna just move in and take it out. Okay, just because this is fun, all those other cells, like neutrophils, macrophages that we talked about as part of the innate immune system, notice since they're always present, right, they're very early on, they're trying to work on this. Macrophages are engulfing those pathogens, trying to destroy them, but also, right, presenting them, showing them to the lymphocytes so that we can crank up these antibodies. Okay? And antibodies then can be very useful in telling us who has been exposed to a pathogen, who's recovered from a pathogen. Anyway, I just find this immune system totally fascinating. So I hope you really enjoy reading about this, thinking about this, and I'm looking forward to talking to you guys more.